All right, let's rock and roll with the nervous system. So this is chapter 12 in the Martini textbook, and this is your first intro to neurons and the various support cells. So mostly what we're covering in this lecture series, and it's going to be more than one because this particular PowerPoint set is like 70 something slides, which is way too many for one video. But to give you a little overview, we will look at the features, cell types, and histology of neural tissue, and then talk about how those cells function. So this is going to include neurons and glial cells, which are support cells, and types of those that are found in both the central nervous system, which is your brain and spinal cord, and your peripheral nervous system, which is everything that's not your brain and spinal cord. So we'll also talk apropos of this about some structural and functional divisions of the nervous system. And of course, we have to talk about both synapses and action potentials. Those are integral to understanding how these cells and tissues do their work, which is to use electricity in the form of ions that flow to control the rest of your body. So let's get into it. So I mentioned divisions. Let's first look at the anatomical divisions. So you're going to hear me say CNS or PNS a lot of times. When I say that, what I mean is central nervous system, which is literally just your brain and your spinal cord, nothing else. So no nerves that carry any of that information away or to, just those structures, which are illustrated here in bluish. And then in the peripheral nervous system, we've got, let's not use red, that's an angry color. How about blue? Peripheral is literally everything else. So all of this stuff, sensory organs, motor neurons, etc., so forth. So all of your nerves. So this is an anatomical division, but there's also some functional divisions as far as which things are sending and receiving parts and which things are processing parts. So the CNS is processing, which you can also think of as computing. So your computing power, your computational ability lives in your brain and to a lesser extent your spinal cord. And then the PNS is mostly about sending and receiving. So sending motor information to control various parts of you or receiving sensory information and sending that sensory information up to the CNS. So that's kind of the purpose-based division of these two anatomical divisions. So in overview, more on that topic, we can see there's additional divisions as you go down. So I already addressed the information processing or computing power aspect of the brain and the spinal cord. And then I said sending and receiving. So you'll note that within the peripheral nervous system block, which in this case is yellow, we've got these words, afferent and efferent. So afferent means toward, I need to use a smaller dot or write larger. Let's do this again. Toward, there we go. So any information that is traveling towards the CNS is called afferent, and any information that is issuing away from CNS is efferent. So notice up here, we see sensory and motor. So let me just blank out the screen real quick and I will describe to you how you can memorize these, because there's a trick to it, and anytime we can use memory tricks to help us memorize stuff to make our lives easier, that's good. So here is the mnemonic device. Same. Sensory equals afferent, so SA. and then motor equals efferent. So that's ME. And there's a second half of that mnemonic device, Dave, same Dave, but we'll get to that when we get to the spinal cord because it's the Dave portion is specific to the spinal cord. Okay, so back to our little PowerPoint here. So you can see sending and receiving motor and sensory information. And then down here we have the division of effectors and receptors. So receptors are basically sensors, and effectors are all of these things. So one thing it would be a good idea to look at is be able to list all of the various kinds of effectors. So notice all three kinds of muscle tissue, glands, and fat. Those are the effectors. And then we'll get into sensory receptors a little bit more later, but basically your special senses are your quote-unquote 5 senses, and then these include touch, but also internal senses that don't rise to your conscious attention. So things like, what's your blood pH like? Is it too acidic? Is it too basic? You can't detect that by yourself, but your brain sure knows about it. So those are senses. So that's just to say, just because something doesn't rise to your conscious attention doesn't mean it's not being detected by your body. It is. Okay, so functional divisions. So notice we have afferent, sensory, efferent, motor again. So just to recapitulate, because repetition is our friend when integrating new information. Incoming information, when I say incoming, I mean incoming to CNS, and outgoing means from CNS. So in the afferent division, which is bringing information into the central nervous system so that that information can be processed or computed, it's mostly information from receptors, which there's super duper lots of types. Interoreceptors, chemoreceptors, mechanoreceptors, we'll talk about a lot of different kinds. And these are located in organs and tissues. The impulses from those receptors are carried to the CNS in the form of sensory neurons, which are typically unipolar. So that word shouldn't mean anything to you yet. I'm giving you a preview because one of the things that we'll do in this presentation is also talk about the structural types of neurons. So not all neurons are created equal. They have fun shapes that they come in that allow them to do some special stuff. And so a lot of your sensory neurons are unipolar. So that's just a preview of some shape info to come. So in the motor division, information is going to flow from the CNS to motor neurons. And you're probably now used to thinking of motor neurons as being only to skeletal muscle, but there's lots of other effectors. So remember all the other kinds of muscle tissue, both cardiac and smooth, and glands and fat. So motor neurons are not only for skeletal muscle, they're also for other stuff. Okay, so within the motor division, we can divide that division by the effectors that it affects. So the somatic, soma means body, which doesn't really do us much good to know that here because there's really no relationship mentally between body and skeletal muscle, but whatever. So the somatic nervous system basically is motor output to skeletal muscle only. So this is your voluntary control of your skeletal muscles. The autonomic nervous system is everything else. And this side is, for the most part, voluntary, and this side is involuntary. So motor output that is taken care of automatically by your autonomic nervous system. So one way to remember kind of some details about the autonomic nervous system is you can think autonomic sounds a lot like automatic. So these are processes that contribute to homeostasis and help keep you alive, but not ones that you ever have to consciously think about. They just happen because your autonomic nervous system takes care of that stuff for you without you having to worry about it. So you don't have to sit around and be like, is my cardiac output sufficient for my activity level? Nope, let me just raise my heart rate a teensy bit. Or let me just go ahead and make my blood vessels a little bit narrower to increase my blood pressure. We don't have to think about that stuff. Our autonomic nervous system does it for us automatically. Okay, so you've seen neural tissue by now. Hello. My dog is trying to help. This is not something you can help with. You're a dog. You don't know any of this stuff. And yet he stares at me as though I'm offending him. So, neural tissue consists of neurons. You guys saw a slide that looked very like this, where there was a nervous whole mount. They're typically taken from oxtail spinal cords. And you can see the cell bodies of neurons here. And then all these little peppery things are the nuclei of glial cells, which are supporting cells. So we're going to look at the kinds of neurons and the kinds of glial cells and talk about their functions. And then we'll start narrowing down to things like action potentials and synapses. So neurons are the major functional unit of the nervous system. They vary in form, meaning there's lots of different shapes. So I mentioned unipolar earlier for sensory. This kind of neuron is multipolar. That's the shape name for this appearance of a neuron. So we'll get into that a little bit more later, but I just want to keep giving you previews. So they vary in form. They also vary in location. There's some in the CNS, there's some in the PNS. Which shapes they take a little bit depend on whether they're in the CNS or the PNS. And for the most part, they are amitotic, meaning no mitosis. So what that means is they're not going to divide and replace themselves. So this is one of the reasons why it's difficult to recover from nerve damage and nervous tissue damage, because these cells are not very regenerative. Most of them do not replace themselves, so it takes a long time for nervous system damage to heal, if it heals at all. So be kind to your nervous system. So with regard to neurons, there's a couple parts that you already know from lab. So let's just build up from there. So this main part, oops, let's do this in a different color because blue is not very visible. So this main part that houses the nucleus is called the soma or neurosoma. So that just means the cell body. Coming off of the cell body are a variety of extensions and projections. So in this picture, these two pictures actually, the neuron in the picture is multipolar, which means it's going to have lots of dendrites, which are these shorter branching processes. And dendra, that root means tree. So the dendrites are named because of their resemblance to trees. Hence, dendrite or dendritic. The area of the cell body that is around the nucleus, so the cytoplasm around the nucleus, is called the perikaryon, and then the nucleus plus the perikaryon makes up the cell body. Oh no, my alarm's going off. My alarm went off to remind me of something that I don't need to do anymore. So that was fun. So, anywho, as I was saying, the neuron cell body is also called the soma. So you might hear me say that word. I mean cell body, which includes these two things. And then there is only one axon. So the dendrites are the receiving end of the neuron, So that's where other neurons interact with this neuron to tell it stuff. And then this neuron has only one sending part, where it sends information to other neurons or effectors, etc. And so this one part is called the axon. So there's one axon per neuron. Now, that axon may split into an axon collateral, which is a branch of the initial axon, but it's still only considered to be one axon because there's only one origination point off of the cell body, and that origination point is called the axon hillock. Hillock is a word that means little hill, and indeed it looks like a little hill. And after that, there's a narrowing, which is the initial segment of the axon, and then it's axon proper. So even if there is a branch in the middle of the axon, it's still only considered to be one axon. Here's why. If I initiate an action potential here, it will guaranteed travel all the way to the end of this axon and any of its collaterals. There is no way to, for example, only have the action potential go this way and not have it go this way. So, because of its electrical behavior, it's considered to be one axon. So, axoplasmic transport refers to the transport of materials from the soma down to the end. So anterograde and retrograde. So anterograde means toward axon terminus or end, and retrograde means away from axon terminus, so towards the cell body. So this is not the same thing as an action potential, it's different. This is how you would, for example, use the genes in the nucleus to transcribe and translate the proteins that make up neurotransmitters. And then you need to carry the neurotransmitters because this is where neurotransmitters are released from. But this is where neurotransmitters are made. So NTs made here. NTs released from here. So they have to get there somehow. And the way they get there is axoplasmic transport, which is really cool. Oops, I didn't mean to do that. So I'm just going to give you a little breakdown of axoplasmic transport really quick, just because it's really cool. So I'm going to draw a really big axon, and then we'll draw the part of the cell body as well. Actually, I want to make this even wider, I think. Okay, so I'm not going to include the axon terminus in this picture, but I'm going to say it's that direction. So we have microtubules, which I'm going to make be blue dots. Make them a little bigger. So inside of the axon, in the axoplasm, which is the cytoplasm that's inside of the axon, because it is a tube of membrane, so it has to have cytoplasm in it too, there's a bunch of microtubules. There we go. There's our tubules. Okay, cool. So let's label those. So these provide a road down which you can transport little packets of neurotransmitter. So, hold on just one second. Okay, I'm back. I just had to silence another alarm. Oops, I missed a little, there's a hole in my microtubule. There we go. So, if we know, let's draw ourselves a nucleus. So nucleus lives over here. And then I'm going to have some endoplasmic reticulum, which you might remember is a place where ribosomes live. So let's draw some of those. And ribosomes are going to make the mRNA into protein via the process of translation. And then we also have to add, oh, let's make this be different color just because I want to make sure that we're differentiating. Okay. The Golgi apparatus, which looks like a little stack of pancakes. So, neurotransmitters are made over here. The nucleus contains the instructions for them. They are transcribed and translated by the rough endoplasmic reticulum, and then they are packaged into vesicles by the Golgi apparatus. So what the Golgi apparatus is going to do is basically release little membrane-enclosed vesicles full of neurotransmitter. So let's label those guys. And we have to get those vesicles down to the end of the neuron somehow. They won't go there by themselves because they're not smart. They don't know anything. They're just a little bubble of membrane with neurotransmitter inside. Oh, and let's label our Golgi apparatus also. So this begs the question, how do they get there then? And the answer is a little motor protein walks them down there. So it's shaped kind of like myosin. It has two little feet and then a tail. So it attaches to the bubble of membrane, and then its feet walk end over end. So it takes little steps down this microtubule. So this motor protein is called kinesin. So what it does is it uses ATP to shift over its little foot, and then it lets go of the cleaved ATP, steps down onto the microtubule, and then does the same thing again with its back foot. So it literally walks the vesicle down the microtubule. So this is anterograde axoplasmic transport. Sometimes we need to bring vesicles back from the periphery, so back from the axon terminus. Who knows why? Maybe we need to recycle something. Doesn't matter. Doesn't matter what it is. Maybe it's some membrane. Maybe there's some receptors in there. We got to recycle these receptors in this membrane because those fats and proteins are useful, the phospholipids and the proteins that make up organelles and membranes and whatnot. So we're going to walk those back using a different motor protein called dynein, and it does the same thing. So it walks in the same manner, but it walks in the opposite direction. So let's label that. Dynein performs retrograde axoplasmic transport. So moving things away from the end of the cell towards the cell body. So the take-home message here is that axoplasmic transport is contingent upon having a road, microtubules, along which little motor proteins named kinesin and dynein walk. And kinesin and dynein walk using ATP energy. So in addition to all of the membrane-based stuff that your neurons are doing, so graded potentials, action potentials, all of those difficult to understand things that you'll come to understand better and better the more we interact with them, ATP is also being used to move and ship materials along axons. So, from this perspective, it makes a lot of sense that neurons in particular are very greedy for glucose because they need a lot of glucose to make a lot of ATP. They're very busy little cells. So I like the way this turned out. I think this looks good. So actually, you know what? When I like things like this, I try to take a screenshot of them so that I can save the screenshot for you. So that is what I will do now. Hello, screenshot, please. Sometimes it doesn't work immediately. There. Well, hope that worked. Okay, so let's get rid of that. Alrighty, so that is what I mean by axoplasmic transport. So with that, let's move along, and we're going to talk about structural types. So this is what I was talking about when I mentioned the shapes that neurons can be. So anaxonic means only dendrites. It's a weird kind. They're not especially common. So an example of where you can find these is in the adrenal medulla. So the middle part, inner part of your adrenal gland, and that is where epinephrine and norepinephrine come from, so adrenaline. And you might be wondering, how can you tell it's a neuron if there's no axon? And the answer is there are cell surface markers, so proteins and such, that are specific to neurons and only neurons. And if you put them on these cells and they stick, that means that this cell is a neuron even though it doesn't have any axon. So that's how we know. So bipolar neurons have two processes separated by the cell body. So on one side we have an axon, and on the other side we have a dendrite, but in this case, instead of being at the end, the cell body is in the middle. So these are next rarest. So they're a little bit more common. Let's put it that way. But not by much. So these tend to be specific to sensory structures. So for example, you have a lot of these in your retina of your eye. Unipolar neurons have one long axon with a cell body that hangs off the side on a stalk like a little lollipop. And then the dendrites just stick off the end of the axon. So this is the receiving end, remember, so the initial segment of the axon is here, so the dendrites are able to directly transfer any excitation they receive right to the axon. They skip the cell body entirely. So these are a little bit more common. And a place where you would find these is peripheral sensory neurons. So the neurons that are going from receptors in various places and traveling via nerves into the spinal cord to talk to the CNS are this kind of nerve. And then multipolar neurons are the most common. So these are all over the place. Examples are motor neurons, both of the autonomic and somatic nervous system, and the neurons that make up your cerebral cortex, which is the outer part of your brain that's made of gray matter, and it is your thinky bits, so the part that does all the processing and computing. So what you'll be expected to do is recognize these neurons by their shape and be able to say whether they are bipolar, unipolar, or anaxonic. So here's just a recap of places you can find these. And I want to put a little asterisk on poorly understood. Research goes on. And the more we research, the more we understand them. So there are some in the brain and sense organs as well. So for example, in the sense organs, there's some in the eye that are also in the retina. They appear to coordinate the activities of the other cells in there. but we don't understand the anaxonic ones quite as well as we do the rest, but we're learning more and more. So research is how we move forward. So for functional classification of neurons, it's a little bit easier because we just divide them into sensory, motor, or inter. So sensory and motor are pretty easy to understand. Interneurons, these are neurons which facilitate communication between other neurons. So neuron A tells interneuron to tell neuron B to do something, for example. So interneurons are critical in your cerebral cortex again, and also for coordination of things like reflexes, so spinal reflexes, which we will discuss, and a variety of other things. So they'll keep popping up. So sensory neurons, and let's just get all these out here, come in two varieties, and those are somatic sensory and visceral sensory. So we talked about the somatic and autonomic nervous systems as far as their motor control and which effectors are affected, but we didn't talk about the sensory aspect of that. So somatic senses are things like proprioceptors, so provide information about your body position. So what is my arm doing? Is it raised in the air? Am I activating my muscles? How is my joint feeling? Is it stable? Is it not stable? Those sorts of things. And then visceral senses are mostly things that don't rise to your conscious attention, with the exception of visceral pain. But, the specific pH of your stomach at this very moment, not something that you could probably put a number to, not something that you're actively sensing, but your brain knows about it and it's being sensed because that pH is going to change depending on your acid secretion, adding or subtracting food, etc. So that's an example of a visceral sense, something that you would never think about unless I told you about it, like I did just now, but which is nonetheless sensed by your nervous system. So these begin at a receptor, which remember there's a lot of different kinds of these. We will discuss a handful of them, huge variety, and they end in the central nervous system. Motor neurons also come in two flavors, somatic or visceral. And these are a little bit easier to understand because this is the motor division. So these begin in the CNS and end at effectors. So somatic motor. Ooh, when I hold my computer like that, I have terrible handwriting. Let's try that again. And visceral motor. All other effectors. So these begin in the CNS, so that means that their soma and their dendrites are in the CNS, and they end at effectors, so they extend out from the CNS and they end at the PNS, which are things like these. So let's practice naming the 5 effectors again. Skeletal muscle, smooth muscle, cardiac muscle, so all of the muscle types, fat, which is adipose tissue, and generally glands. What kinds of glands, you might be wondering? All kinds, many kinds. We will discuss more about those later. So, motor, efferent, go out to affect effectors. And finally, interneurons, which are also called association neurons. These are what I said they were. So basically, they are going to be allowing two neurons that are not the interneuron to talk to each other. So they're often in the context of reflexes, for example, between sensory and motor neurons, but there's also plenty in the cerebral cortex, for example, that are not between sensory and motor neurons. So this is a case of often, but not always. So you have intricate pools and circuits of neurons with lots of interneurons that are all in your cerebral cortex, and they allow you to do things like remember stuff, plan things, learn, form opinions, have intentions. And none of those are interneurons that are between a sensory and a motor neuron necessarily. However, they may require sensory input and motor output sometimes, but again, often, but not always. In most of biology, there's rules, and then there's numerous exceptions to the rule. That's one of the reasons biology is difficult. So I'm just going to do a quick time check on this video. And I actually think we're going to end it there because the video will upload more quickly if I keep them shorter. So I'm going to pause this here. So when we come back in the next nervous system video, we'll start talking about neuroglia, which are those support cells, and why they're shaped weird, what they do, and how they're divided, because they are divided into central and peripheral in a similar manner to the neurons. So that's a preview of what you can look forward to in the next video. So I'm going to get to posting this, and I will see you guys later.